So let's talk more about glycolysis. So this is the first phase of cellular respiration, and just as a reminder, it takes place in the cytoplasm, meaning it takes place outside of the mitochondria. It is an aerobic process, meaning it doesn't require any oxygen, and it's also going to serve as the foundation for both aerobic and anaerobic respiration. And glycolysis is named so because we are literally lysing a glucose molecule in half. Glucose is a 6-carbon molecule, and we're going to lyse it in half to produce 2 3-carbon molecules, and those 3-carbon molecules are referred to as pyruvate. It is about a 10-step process in order to get from glucose to pyruvate. You don't need to know all the individual steps, but you should be able to recognize that it is divided into 2 main phases and what those phases entail. So we have an energy investment phase and we have an energy harvesting phase. So in the energy investment phase, we're going to actually use 2 ATP in order to get this system started, in order to begin the breakdown of glucose. So the energy that we're using is going to come from 2 ATP molecules. And these 2 ATP molecules are going to add phosphate groups to the end of the glucose molecule as represented here. And this phosphorylation is going to help prime glucose and convert it into a more reactive form, which is eventually going to lead to the formation of something called glyceraldehyde 3-phosphate, or G3P. One of the intermediate products formed during this phase is 3-phosphoglycerate, or 3PG, and another one is 1,3-bisphosphoglycerate, or BPG. These are just high energy intermediates, and they're essential in facilitating the next steps where we're actually going to harvest energy. So the other phase of glycolysis is the energy harvesting phase. So we had to input 2 ATP in order to prime the system and to get it started. Now we need to actually get some energy out of it, because that's the whole purpose of cellular respiration and glycolysis. And this is going to involve the oxidation of G3P and lead to the production of 4 ATP molecules through a process called substrate level phosphorylation, which means that phosphate groups were directly transferred from a substrate from ADP in order to form ATP. Since 2 ATP molecules were consumed in the earlier phase, we consider the net gain from glycolysis to be 2 ATP per glucose molecule, because remember, it cost us 2 to get it started. We produced 4, so we net a gain of 2 ATP molecules. We're also going to get 2 molecules of NAD, which are going to be reduced to NADH, and later we're going to use those at the electron transport chain to generate more ATP during aerobic respiration, assuming that we have oxygen available. So at the end of glycolysis, the fate of pyruvate molecules is really going to depend on, do we have oxygen available or not. If oxygen is present, pyruvate is then going to enter into the mitochondria to be further processed through the preparatory reaction in the citric acid cycle, and then eventually lead to the electron transport chain. However, if we don't have oxygen, the cells are going to turn to fermentation, which is an anaerobic pathway, to continue to generate ATP and regenerate NAD, which is essential to keep glycolysis running. So if we don't have any oxygen available, again, we're turning to fermentation and anaerobic pathway in order to make ATP and regenerate NAD+. So when we talk about fermentation, it's really the incomplete breakdown of glucose, and it's going to occur entirely in the cytoplasm without the involvement of the mitochondria at all. So it's going to result in the production of only 2 ATP per glucose molecule, making it far less efficient than aerobic respiration. If we're talking about animal cells, we do have cells even in our human body that are capable of undergoing fermentation. One of it is our skeletal muscle cells, and it's going to lead to the conversion of pyruvate into lactate, which is lactic acid. So if you've ever been exercising vigorously and you feel that burn in your thighs or your arms, it's from lactic acid buildup, meaning that your cells were not getting enough oxygen at that point. However, this process is going to provide a quick burst of energy, especially useful during those intense muscular activities when the oxygen supply is limited. However, lactate accumulation can also contribute to muscle fatigue. So once adequate oxygen is restored, a state known as recovery from oxygen deficit, we're going to take lactate, transport it to the liver, and convert it back to pyruvate for use in aerobic respiration, or recycle it into glucose via a different cycle, which we're not going to talk about. So, in contrast, we have microorganisms that can employ a variety of fermentation pathways depending on their species and environmental conditions. So, for example, certain bacteria can produce lactate, like we just talked about, or other organic acids as end products. Others, such as yeast, they can carry out a different type of fermentation called alcoholic fermentation, where they're going to convert pyruvate into ethanol and carbon dioxide. And this form of fermentation has widespread applications in food and beverage production, especially if we're talking about bread making. The carbon dioxide that's produced by yeast is what's going to cause the dough to rise. And we're also going to have ethanol that can contribute to the production of wine and beer. So glycolysis is really important. It serves as a universal starting point for glucose metabolism, whether oxygen is available or not. Again, if oxygen is scarce, fermentation pathways are going to make sure we still have ATP being produced. And this evolutionary adaptation has really allowed cells and organisms to survive in anaerobic conditions, even if only for short periods.